Welcome students and welcome to my channel Sequential Biology, and we are talking about reproductive strategy of different type of viruses, and already we have discussed about reproduction strategy of double-stranded DNA containing viruses, single-stranded RNA containing viruses and the double-stranded RNA containing viruses. And today we will discuss about reproductive strategy of retroviruses. This is a structure of a retrovirus, and the retrovirus are usually enveloped virus. So this is enveloped, and also inside envelope there are present capsid. So this is capsid layer, okay, and in the envelope there are present many glycoprotein, and this acts as spike protein and which binds with a receptor of host cell. And also in case of retrovirus there are present different type of enzymes. Since this is HIV virus, so there are present two important enzymes, the integrase and the reverse transcriptase, and this reverse transcriptase enzyme was first discovered by David Baltimore. HIV and other retroviruses, two copies of non-segmented plus ssRNA. So in retroviruses there are present two copy of single-stranded RNA, but the single-stranded RNA are plus-stranded. Because they are the plus-stranded, so they can directly involved in translation, right? So non-segmented plus ssRNA and enzyme reverse transcriptase and integrase are present inside these retroviruses, and these enzymes are utilized during their replication. How retrovirus reproduce? And you know that retroviruses have two copies of RNA, and these two copies of RNA are obviously the single-stranded RNA, but they are plus-stranded RNA. Plus-stranded means this strand directly involved in protein synthesis. And the reverse transcriptase enzyme are those enzymes which can synthesize DNA from RNA. Transcriptase is RNA-dependent DNA polymerase. Then we know that the DNA have the two complementary strand of DNA. One is 5' to 3' orientation. One strand orientation is 5' to 3', and another strand orientation is 3' to 5', and this 3' to 5' strand is served as template, okay, and from this template strand generally the mRNA are synthesized by using enzyme RNA polymerase, okay, by using RNA polymerase. So synthesis of RNA from DNA, so synthesis of RNA from DNA is called as transcription, okay, so synthesis of RNA from DNA is called as transcription. But the synthesis of DNA from RNA, is called as reverse transcription. This is known as reverse transcription. And reverse transcription is occurred by a special type of enzyme which is known as reverse transcriptase. Plus ssRNA, with the help of the reverse transcriptase enzyme, from this plus ssRNA a single strand of DNA is synthesized, and this DNA strand must be complementary with this RNA, okay. So first step in their replication is the synthesis of a RNA is to DNA hybrid. Then this is RNA chain, and this is DNA strand and DNA chain, and this RNA DNA hybrid first synthesized. The reverse transcriptase enzyme show the ribonuclease activity, so the RNA will be digested or RNA will be broken, and this will release a single-stranded DNA, okay. So this single-strand DNA will be released from this RNA DNA hybrid. Next, the DNA polymerase actually causes the replication of this single-stranded DNA, and this is the process by which the double-stranded DNA is synthesized, right? Now this double-stranded DNA, which is usually synthesized from this single-stranded RNA, are transported into the host cell nucleus, and inside the host cell nucleus they will be incorporated into host cell DNA. Then this is host cell DNA, and inside this host cell DNA this viral double-stranded DNA will be incorporated. The incorporation inside this host cell DNA, the synthesis of this double-stranded DNA or formation of this double-stranded DNA will be very, very important. This integrase enzyme is present in retrovirus, in every kind of retroviruses, and this integrase, the function of this integrase enzyme is to incorporate of this double-stranded DNA into the host cell DNA. Transcription by the host cell RNA polymerase, since this is a double-stranded DNA, and they will follow the normal transcription process, and they will form the mini mRNA, okay, and they use the host cell RNA polymerase, and also with the help of host cell RNA polymerase the single-stranded RNA will be produced, okay. This hybrid DNA, suppose this is a chimeric DNA, and one is 5' to 3' strand, and this DNA have two strand, the 5' to 3' strand and 3' to 5' strand. And this 3' to 5' strand, when transcribed, this will form plus RNA. Okay, and when this 5' to 3' strand will transcribe, this will form the minus RNA, right? Now the 3' to 5' strand will be transcribed to form the mRNA, that is the plus RNA, and also the host cell polymerase will produce the plus ssRNA, and this is the viral ssRNA. And by the process of translation, this mRNA will be translated and to form the viral protein, and this viral protein and the plus ssRNA will be assembled together, and this will form the daughter virion. Okay, so inside this daughter virion we can see the two strand of ssRNA. After entry of plus ssRNA, reverse transcribed to form the ssDNA, single stranded DNA, by viral reverse transcriptase, which from RNA is to DNA hybrid. The reverse transcriptase also have ribonuclease activity. This is also an important activity of reverse transcriptase. This will digest the RNA from the RNA DNA hybrid. Okay, so reverse transcriptase actually digest this RNA from this RNA DNA hybrid. Okay, and this will causes the release of this single stranded DNA. Now resulting the single stranded DNA, and this will converted into double stranded DNA by DNA polymerase activity of reverse transcriptase. Then the reverse transcriptase is very important enzyme, and they are involved in three steps. First, this single stranded RNA plus ssRNA, from this plus ssRNA they will synthesize a DNA, and which is complementary with this plus ssRNA. And second, they will causes the digestion of this RNA, and the third, they have the DNA polymerase activity, and from this DNA the single stranded DNA will be converted into the double stranded DNA. Then this reverse transcriptase enzyme is very important for their reproduction. Okay, the double stranded DNA then transported into the host cell nucleus, and inside host cell nucleus we know that the transcription process is usually performed inside nucleus. So this double stranded DNA must be transported into host cell nucleus, and the viral double stranded DNA integrated into host cell DNA by the viral integrase. The integrated DNA serves as template. Obviously this integrated DNA serves as template for production of plus ssRNA and the viral structural protein, and this plus ssRNA, many plus ssRNA will be synthesized, and also the viral structural protein will be synthesized, and this plus ssRNA and the viral structural protein will be packaged together, and they will form a complete virus particle. In next classes we will discuss about the structure of tobacco mosaic virus. The tobacco mosaic virus is the first discovered virus, and we will discuss about structure of tobacco mosaic virus.